Hello students, in this part we will talk about structure of DNA. DNA was firstly identified by mixture in pus cell. DNA was firstly identified mixture, but DNA term was coined by Zacchaeus. DNA term was coined by Zacchaeus, Watson, and Crick. Model of DNA. Here it's a first strand. DNA is double helix structure. So second strand is parallel to this. In the first strand, both of these strands are interlinked with nitrogen base hydrogen bonds. Watson and Crick model of DNA. First of all, parallel to this, we just go through with molecular structure of DNA also. These are the pentose sugar which are interlinked with phosphate group. Here on third carbon there is a OH group. There is a OH group and here phosphate group. Here the elimination of OH water molecule and is interlinked with the other or second sugar. Such type of structure make the backbone of the DNA molecule or RNA molecule here. On a first carbon there is a nitrogen base. This chain is also interlinked with phosphate group. There is a OH where phosphorus group is attached. CH2 phosphorus, CH2 phosphorus, CH2 phosphorus, CH2 phosphorus, CH2 here. If you observe this structure, then it's a 5' end. 5' end. We just call it as 5' end, and the tail portion where OH group is present or a third portion OH group is present is called 3' end. On the opposite side, it is just reverse. It's a 3' end and a 5' end. Means if one strand is from 5' to 3', then the orientation of other strand is 3' to 5' here. Means both the strands are parallel to each other, but they are anti-parallel. Means they runs through parallel with each other, but in a opposite direction here. If a one strand, we just note it, the one strand nitrogen bases A, G, C, T, T, they are in a random fashion, but the other strand is just parallel or according to the first strand. Means A always bind with T as we did in a previous video. G, C with triple hydrogen bonding, C, G with triple hydrogen bonding, T, A with double hydrogen bonding, and T, A with double hydrogen bonding here. So here two strands, there is a anti-parallel to each other. First is a 5' to 3', then other orientation is 3' to 5' direction. It's a molecular structure of DNA. The other one structure of Watson and Crick is if it's a diameter of the DNA is 20 angstrom. The diameter of DNA is 20 angstrom. The gap where the DNA molecules are closer or the base pair are closer, this one is called minor groove. This part is called major groove. In 360 angle, or one minor to minor or major to major, one minor to other minor or one major to other major, we just calculated it. There is a minor to another minor. There are 10 base pairs. Just calculated here. 1 in between these, 2, 3, 4, 5, 6, 1, 7, 8, 9, 10, and this one counted in the another strand. So in a one ring, one 360 angle, or one minor to minor, one major to major, there are 10 base pairs. The distance between any of the two base pairs is 3.4 angstrom. The distance between any of the two strands is 3.4 angstrom, and distance between one ring is 3.4 multiplied by 10, it's 34 angstrom. Here A bonded with T and G bonded with C, as the Watson and Crick told. So what are the main point of this? (34 angstrom). The main points of DNA are: first, DNA is negative charge. Why DNA is negative charge? Due to the presence of phosphoric acid. DNA is acidic. DNA is acidic in nature. Third, diameter 20 angstrom. Distance between two base pairs 3.4 angstrom, or between one ring 34 angstrom. Fifth, purine is equal to pyrimidine, means number of A plus G is equal to C plus T. Here 6 point, A always bond with T with double hydrogen bonding, and G always bond with C triple hydrogen bonding. DNA strand strands are anti-parallel to each other if one strand 5 prime to 3 prime direction, other strand 3 prime to 5 prime direction. So that's why they are anti-parallel to each other here. Next is Chargaff rule. Chargaff gave some points regarding to the DNA structure and their molecular basis. First of all, A plus G is equal to C plus T, means adenine is equal to purine is equal to pyrimidine. First. Second is A plus G C plus T is equal to 1 means it's a constant for a particular generation of a particular species. Constant for species. Third is A bond T is more than C bond G. A T is more than C bond G is called AT DNA. Is called AT DNA. And the best example is eukaryotes. Here, fourth one, if AT is, we just represented here with their bonds, C triple bonded G. If AT is lower than C bond G, means in a DNA molecule, if CG bonds are more than AT, then it is called CG DNA. Fifth, TM, melting temperature. We are representing here now melting temperature. The melting temperature is C triple bonded G is always greater than A double bonded T for melting temperature. So what we have to do in the Chargaff rule, first of all Chargaff propose that some consequences regarding to the DNA structure. First is A plus G is equal to C plus T, means number of purines are equivalent to the number of pyrimidines here. A plus G upon C plus T means purine ratio pyrimidine. Purine ratio pyrimidine is equal to a constant for a particular species here. In a DNA sequence, if A double bond T is greater than C triple bond G, then it is called as ATDNA. Means in that type of DNA, AT molecules are more. It's an example of eukaryotes. And if it's a reversible, means C triple bond G are more than A double bond T, then it is called as cgDNA. It's an example of prokaryotic cell. Example, prokaryotic cell. Melting temperature, C triple bond G is greater than A double bond T always, because to break down the triple bond take more energy or acquire more energy to break down such kind of bonds, triple bond, as compared to double bond. So triple bond means we can say prokaryotic DNA get more energy or more temperature for a melting as compared to eukaryotic DNA, as compared to eukaryotic DNA here. Now we will talk about denaturation of DNA. If DNA temperature provides 80 degree to 90 degree Celsius temperature, then hydrogen bonds in between two strands of DNA break down So it's called as denaturation of DNA. Means if this strand of DNA get to be approximately raise the temperature 80 to 90 degrees Celsius, then both the bonds between the DNA, means assuming it's a bond between the two strands, these are the two strands, and it's a hydrogen bond between the nitrogen bases. We provide the temperature more than 80 or 90 degrees Celsius, or approx 80 or 90 degrees Celsius, the bond break down and two strands separate out from each other. Renaturation of DNA. Denaturated DNA at room temperature means 35 to 37 degrees Celsius. Hydrogen bonds rejoined. Hydrogen bonds rejoined. Then the condition is called renaturation of DNA. So what we have to do? Denaturation of DNA and renaturation DNA. If we raise the temperature of the DNA molecule, then they split up, the hydrogen bonds split up and two strands are separate out from each other. It's called denaturation of DNA. And denatured DNA again put at a room temperature or provide a room temperature condition, then they again rejoin, or the hydrogen bond between two nitrogen bases rejoin. The condition is called renaturation of DNA here.